Locomotion and Movement, Chapter 17. Movement is one of the significant features of living beings. Animals and plants exhibit a wide range of movements. Streaming of protoplasm in the unicellular organisms like amoeba is a simple form of movement. Movement of cilia, flagella, and tentacles are shown by many organisms. Human beings can move limbs, jaws, eyelids, tongue, etc. Some of the movement result in a change of place or location. Such voluntary movements are called locomotion. Walking, running, climbing, flying, and swimming are all some forms of locomotory movements. Locomotory structure need not to be different from those affecting other type of movements. For example, in paramecium, cilia helps in movement of food through cytopharynx and in locomotion as well. Hydra can use its tentacles for capturing its prey and also use them for locomotion. We use limbs for change in body posture and locomotion as well. The above observation suggests that movement and locomotion cannot be studied separately. The two may be linked by stating that all locomotions are movements, but all movements are not locomotions. Methods of locomotion performed by animals vary with their habitat and the demand of the situation. However, locomotion is generally for search of food, shelter, mate, suitable breeding grounds, favorable climatic conditions, or to escape from enemies or predators. Type of movement. Cells of the human body exhibit three main types of movement, namely amoeboid, ciliary, and muscular. Some specialized cells in our body, like macrophages and leukocytes in blood, exhibit amoeboid movement. It is affected by pseudopodia formed by the streaming of protoplasm, as in amoeba. Cytoskeletal elements like microfilaments are also involved in the amoeboid movement. Ciliary movement occurs in most of our internal tubular organs, which are lined by ciliated epithelium. The coordinated movements of cilia in the trachea help us in removing dust particles and some of the foreign substance inhaled along with the atmosphere air. Passage of sorry, Passage of ova through the female reproductive tract is also facilitated by ciliary movement. Movement of our limbs, jaws, tongue, etc. require muscular movement. The contractile property of muscles are effectively used for locomotion and other movements by human beings and majority of multicellular organisms. Locomotion requires a perfect coordinated activity of muscular, skeletal, and neural system. In this chapter, you will learn about the type of muscles, their structure, mechanism of their contraction, and important aspects of the skeletal system. Muscle. You have studied in chapter 8 that the cilia and flagella are the outgrowth of the cell membrane. Flagellar movement helps in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and in locomotion of protists like Euglena. Muscle is a specialized tissue of mesodermal origin. About 40 to 50% of the body weight of a human adult is contributed by muscle. They have special properties like excitability, contractility, extensibility, and elasticity. Muscles have been classified using different criteria, namely location, appearance, and nature of regulation of their activities. Based on their location, three types of muscles are identified: skeletal, visceral, and cardiac. Skeletal muscles are closely associated with the skeletal components of the body. They have a strapped appearance under the microscope and hence are called striated muscles. As their activities are under the voluntary control of the nervous system, they are known as voluntary muscles too. They are primarily involved in locomotory actions and change of body postures. Visceral muscles are located in the inner walls of hollow visceral organs of the body like alimentary canal, reproductive tract, etc. They do not exhibit any striation and are smooth in appearance. Hence, they are called smooth muscles, that is, non striated muscles. Their activities are not under the voluntary control of the nervous system and are therefore known as involuntary muscles. They assist, for example, in the transportation of food through the digestive tract and gametes through the genital tract. As the name suggests, cardiac muscles are the muscles of heart. Many cardiac muscle cells assemble in a branching pattern to form a cardiac muscle. Based on appearance, cardiac muscle are striated. They are involuntary in nature as the nervous system does not control their activities directly. Let us examine a skeletal muscle in detail to understand the structure and mechanism of contraction. Each organized skeletal muscle in our body is made of number of muscle bundle or fascicles held together by a common collagenous connective tissue layer called fascia. Each muscle bundle contains a number of muscle fiber that is shown in figure 17.1. Each muscle fiber is lined by the plasma membrane called sarcolemma enclosing the sarcoplasm. Muscle fiber is a syncytium as the sarcoplasm contains many nuclei. The endoplasmic reticulum, that is sarcoplasmic reticulum of the muscle fiber, is the storehouse of calcium ions. A characteristic feature of the muscle fiber is the presence of large number of parallelly arranged filaments in the sarcoplasm called myofilaments or myofibrils. Each myofibril has alternate dark and light bands on it. A detailed study of the myofibril has established that the striated appearance is due to distribution pattern of two important proteins, that is actin and myosin. The light band contains actin and is called I band or isotropic band, whereas the dark band called A or anisotropic band contains myosin. Both the proteins are arranged as a rod-like structure parallel to each other and also to the longitudinal axis of the myofibrils. Actin filaments are thinner as compared to the myosin filaments, hence are commonly called thin and thick filaments respectively. In the center of each I band is an elastic fiber called Z-line which bisect it. The thin filament are attached firmly to the Z-line. The thick filament in the A band are also held together in the middle of this band by a thin fibrous membrane called M-line. The A and I band are arranged alternatively throughout the length of the myofibrils. The portion of the myofibrils between two successive Z lines is considered as the functional unit of contraction and is called sarcomere, that is shown in figure 17.2. In a resting state, the edges of thin filament on either side of thick filament partially overlap the free end of the thick filament, leaving the central part of the thick filament. This central part of thick filament not overlapped by thin filament is called H-zone. These are the diagrams of sarcomere. Structure of contractile proteins. Each actin filament is made up of two F, that is filamentous actins, helically wound to each other. Each F actin is a polymer of monomeric G globular actin. Two filaments of another protein called tropomyosin also run close to the F actins throughout its length. A complex protein troponin is distributed at regular intervals on the tropomyosin. In the resting state, a subunit of troponin masks the active binding site for the myosin on the actin filaments, that is shown in figure 17.3a. Each myosin thick filament is also a polymerized protein. Many monomeric protein called meromyosins, that is shown in figure 17.3b, constitutes one thick filament. Each meromyosin has two important parts, a globular head with a short arm and a tail, the former being called the heavy meromyosin and the later the light meromyosin. The heavy meromyosin component, that is the head and short arm, projects outwards at regular distance and angle from each other from the surface of a polymerized myosin filament and is known as cross arm. The globular head is an active ATPase enzyme and has binding site for ATP and active sites for actin. The given diagram is 17.3a, this is, and 17.3b, this is. Mechanism of muscle contraction. Mechanism of muscle contraction is best explained by sliding filament theory, which states that contraction of muscle fiber takes place by the sliding of the thin filament over the thick filaments. Muscle contraction is initiated by a signal sent by the central nervous system via motor nerves. A motor neuron, along with the muscle fiber connected to it, constitute a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter acetylcholine, which generates an action potential in the sarcolemma. This spreads through the muscle fiber and cause the release of calcium ions into the sarcoplasm. Increase in calcium level leads to binding of calcium with a subunit of troponin on actin filaments and thereby remove the masking of active sites for myosin. Utilizing the energy from ATP hydrolysis, the myosin head now binds to the exposed active sites on actin to form a cross bridge that is shown in figure 17.4. This is the figure 17.4 showing stages in cross bridge formation, rotation of head, and breaking of cross bridge. This pulls the attached actin filament towards the center of A band. The Z line attached to these actins are also pulled inward, thereby causing a shortening of the sarcomere, that is contraction. It is clear from the above steps that during shortening of the muscle, that is contraction, the I bands get reduced whereas the A band retain the length, shown in figure 17.5. The myosin releasing the ADP and phosphate goes back to its relaxed state. A new ATP binds and the cross bridge is broken, that is shown in figure 17.4. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ion are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filaments. This caused the return of Z line back to their original position, that is relaxation. The reaction time of the fiber can vary in different muscles. Repeated activation of the muscles can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscles contain a red colored oxygen storing pigments called myoglobin. Myoglobin content is high in some of the muscles which give a reddish appearance. Such muscles are called the red fibers. These muscles also contain plenty of mitochondria which can utilize the large amount of oxygen stored in them for ATP production. These muscles therefore can also be called aerobic muscles. On the other hand, some of the muscles possess very less quantity of myoglobin and therefore appear pale or whitish. These are the white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy. You have to look through this diagram and remember it for exam purpose. Skeletal system. Skeletal system consists of a framework of bones and few cartilage. This system has significant role in movement shown by the body. Imagine chewing food without jawbone and walking around without the limb bone. Bone and cartilage are specialized connective tissues. The former has a very hard matrix due to calcium salts in it, and the latter has slightly pliable matrix due to chondroitin salt. In human beings, this system is made up of 206 bones and a few cartilages. It is grouped into two principal divisions, the axial and the appendicular skeleton. Axial skeleton comprises 80 bones distributed along the main axis of the body. The skull, vertebral column, sternum, and ribs constitute axial skeleton. The skull, shown in figure 17.6, is composed of two sets of bones, that is cranial and facial, that total to 22 bones. Cranial bones are 8 in number. They form the hard protective outer covering cranium for the brain. The facial region is made up of 14 skeletal elements which form the front part of the skull. A single U-shaped bone called hyoid is present at the base of the buccal cavity. Each middle ear contains three tiny bones, malleus, incus, stapes, collectively called ear ossicles. The skull region articulates with the superior region of the skull region articulates with the superior region of the vertebral column with the help of two occipital condyles. That's why human skull is called dicondylic skull. Our vertebral column, that is shown in figure 17.7, is formed by 26 serially arranged units called vertebrae and is dorsally placed. It extends from the base of the skull and constitutes the main framework of the trunk. Each vertebra has a central hollow portion that is called neural canal through which the spinal cord passes. First vertebra is the atlas and it articulates with the occipital condyles. The vertebral column is differentiated into 7 cervical, 12 thoracic, 5 lumbar, 1 fused sacral, and 1 fused coccygeal regions starting from the skulls. The number of cervical vertebrae are 7 in almost all mammals, including human beings. The vertebral column protects the spinal cord, supports the head, and serves as the point of attachment for the ribs and the musculature of the back. Sternum is a flat bone on the ventral midline of thorax. There are 12 pair of ribs. Each rib is a thin flat bone connected dorsally to the vertebral column and ventrally to the sternum. It has two articulation surfaces on its dorsal end and is hence called bicephalic. First 7 pairs of ribs are called proribs. Dorsally, they are attached to the thoracic vertebrae and ventrally connected to the sternum with the help of hyaline cartilage. The 8th, ninth, and 10th pair of ribs do not articulate directly with the sternum but join the 7th rib with the help of hyaline cartilage. These are called vertebrochondral ribs, that is false ribs. Last two pairs, 11th and 12th of the ribs, are not connected ventrally and are therefore called floating ribs. Thoracic vertebrae, ribs, and sternum together form the rib cage. The bones of the limb, along with their girdles, constitutes the appendicular skeleton. Each limb is made up of 30 bones. The bone of the hand for limb are just just look these diagrams. The bone of the hand forelimb are humerus, radius, and ulna, carpals, that is wrist bones, that is 8 in number, metacarpals, palm bones, that is 5 in number, and phalanges, digits, 14 in number, shown in figure 17.9. Femur, that is thigh bone, the longest bone of our body, tibia, fibula, tarsals, that is ankle bones, that is 7 in number, metatarsals, that is 5 in number, and phalanges, the digits 14 in number, are the bone of the legs, hind limb. Figure 17.10 showing it. A cup-shaped bone called patella cover the knee vertically, that is called as kneecap. Pectoral and pelvic Pectoral and pelvic girdle bones helps in the articulation of the upper and lower limb respectively with the axial skeleton. Each girdle is formed of two halves. Each half of a pectoral girdle consists of a clavicle and a scapula, shown in figure 17.9. Scapula is a large triangular flat bone situated in the dorsal part of the thorax between the second and the seventh rib. The dorsal flat triangular body of scapula has a slightly elevated ridge called the spine, which projects as a flat expanded process called the acromion. The clavicle articulates with this. Below the acromion is a depression called the glenoid cavity, which articulates with the head of the humerus to form the shoulder joint. Each clavicle is a long slender bone with two curvature. This bone is commonly called the collarbone. Pelvic girdle consists of two coxal bones, that is shown in figure 17.10. Each coxal bone is formed by the fusion of three bones, ilium, ischium, and pubis. At the point of fusion of the above bones is a cavity called acetabulum, to which the thigh bone articulates. The two halves of the pelvic girdle meet ventrally to form the pubic symphysis containing fibrous cartilage. It is figure 17.10 and it is figure 17.9. Joints. Joints are essential for all types of movements involving the bony part of the body. Locomotory movement are no exception to this. Joints are point of contacts between bones or between bones and cartilage. Force generated by muscle is used to carry out movement through joints, where the joint acts as fulcrum. The movability of these joints, the movability at these joints vary depending on different factors. Joints have been classified into three major structural forms, namely fibrous, cartilaginous, and synovial. Fibrous joints do not allow any movement. This type of joint is shown by the flat skull bone, which fuse end to end with the help of dense fibrous connective tissue in the form of sutures to form the cranium. In cartilaginous joints, the bones involved are joined together with the help of cartilage. The joints between the adjacent vertebrae in the vertebral column is of this pattern and it permits limited movement. Synovial joints are characterized by the presence of fluid-filled synovial cavity between the articulating surface of two bones. Such an arrangement allows considerable movement. These joints helps in locomotion and many other movement. Ball and socket joint between humerus and pectoral girdle. Hinge joint in knee joint, pivot joint between atlas and axis, gliding joint between the carpals, and saddle joint between the carpal and metacarpal of thumb are some examples. Disorder of muscular and skeletal system. Myasthenia gravis, autoimmune disorder affecting neuromuscular junction, leading to fatigue, weakening, and paralysis of skeletal muscle. Second, muscular dystrophy, progressive degeneration of skeletal muscle, mostly due to genetic disorder. Third, tetany, rapid spasm, wild contractions in muscle due to low calcium level in body fluids. Fourth, arthritis, inflammation of joints. Fifth, osteoporosis, age-related disorder characterized by decreased bone mass and increased chance of fractures. Decreased level of estrogen is a common cause. Sixth, gout, inflammation of joints due to accumulation of uric acid crystals. Muscle fiber is the anatomical unit of muscle. Actin and myosins are polymerized protein with contractility. Thank you.